Welcome to Datta Biology classes. Our last episode was a discussion on DNA, the structure of DNA and others. We have learned. Now we'll learn how this DNA replicates. So why it is required, where it is required, and what is actual definition of DNA replication we'll learn today, one by one. So let us start with the definition first. DNA replication. DNA is, this is the hereditary material, or DNA is polynucleotide chain. So within the cell there is chromosome, nucleus. A nucleus contains chromosome. This is the cell. Cell contains a nucleus. Nucleus contains chromosomes, and these chromosomes, within this chromosome, the genes are located, or DNA. Genes are made up of DNA. DNA are there. So, in definition, we can write that DNA replication is a process by which a cell replicates or cell make copies of its DNA. A cell make copies of its DNA. Copies of its DNA. Or we can say a cell make identical copies. Identical copies of its DNA. So, the DNA replication, we can say DNA replication is a replication of the DNA. That will not serve the purpose. We will say the replication is a process occurs in a cell in which or by which or through which the cell replicates or the cell make identical copies of its DNA. Why it is required, why it occurs, will come. Let us start that whenever we have come across mitosis and meiosis. Mitosis and meiosis. Mitosis occurs in somatic cells and meiosis occurs in reproductive cells. These cells, all the cells, maybe somatic cells or maybe reproductive cells, they undergoes through these two divisions. In mitosis, two cells are produced. Or in meiosis, there are 4 haploid cells are produced. How it is produced? Because we know that there are some stages known as prophase, metaphase, anaphase and telophase. We have gone through this prophase, metaphase, anaphase, telophase. After the telophase, cytokinesis occurs, in which cytoplasm, so prophase, metaphase, anaphase, telophase, then cytoplasm divides, then 2 cells is produced. 2 identical cells are produced. After the cytokinesis, the cell takes rest. Those stage is known as interphase. That is, the interphase consists of gap 1, gap 2 and S phase. We have gone through. So after the cell division is over, after cytokinesis, there is a gap. It varies from species to species. So after the mitosis is over, the cell goes through interphase. In the interphase, first the cell takes a gap, that is rest, and thereafter that cell synthesizes its DNA. We have gone through. And then again a gap, and then the cell becomes ready for next division. Okay. So mitosis occurs. Why mitosis occurs? DNA replication is a process by which the cell makes identical copies of its DNA. It is required for growth, repair. So the cell may it be plant cell, may it be animal cell. So it requires growth and it requires repairing of its cell. Sometimes the cell get lost by there is an accident or something like that. So the cell reproduce. So for growth and repair, the cell requires mitosis, cell division. Even in mitosis, synthesis phase, these new cells are produced, new DNA are produced. Again, I told you that why DNA replication requires? Requires for growth, repair, and this is very much important, and reproduction. And for reproduction, that is meiosis. So in meiosis, what we have learned in our classes, please go to my earlier classes before 1 or 2 months, mitosis or meiosis. There you will find that in meiosis there is mitosis also, that is called meiotic mitosis. So after mitosis there is meiosis. One is reductional division, another is equational division. But there also it undergoes through synthesis phase. In that phase, for reproduction, the new synthesis of DNA takes place. That is why this DNA replication is so important. You consider, let us consider that I myself was born as a single cell, having 46 pair of chromosomes. Okay, this cell contains chromosomes. So those chromosomes underwent so many multiplication division, that is mitosis for my growth, because I was born as a single cell and then I have come to millions and millions of cells in my body. So I went through mitosis. Again, I have married, I have given birth to some other organisms, other species of my own. So in that case, I went through my cell. Reproductive cells went through meiosis. So what I will say, that once I was born as a single cell. It is a single cell having a single nucleus. And in single nucleus, nucleolus were there and chromosomes were there, 46 chromosomes, that is 26 pairs. And then whenever I developed by mitosis as well as meiosis, the cells went through various stages. Those stages are called blastula, gastrula, morula, so many embryonic development went on. And the irony is that after some time, some cells has become meiocytes, that is for meiosis. Some cell has become other somatic cells. And say for example, its DNA, the DNA inside this cell have the power to produce bones. So the production of bones is in the DNA. So that DNA produces the bone. That DNA produces my eyes. That is, eye cells are produced, differentiated. Their DNA produces nerve cells or neurons. So there was a stage after so many mitosis, there was a stage when this differentiation, functional differentiation occurs. Those cells, some cell has gone to produce nerve cell, some cell has gone to produce bone cell, some cells have produced eye cell. So there was a particular point of cell division where from differentiation, functional differentiation, and the cell divides. So these individual cells which had this DNA had all the characteristics of producing brain, producing heart muscles, so many things, muscles and bones and everything. So the cells are sometimes called totipotent. That is, it has the ability to produce. Sometimes there was a saying that once nerve cells, once we meet an accident and scooter and other without having helmet, my brain cells is lost. Once the brain cells, that is neuron, is lost, it cannot be repaired, it cannot be regenerated. Why? I should request all of you that you think that particular point of time where from this cell has differentiated itself into a nerve cell, this cell has differentiated into eye cell, this cell has differentiated into heart muscle cell. So that point, if you find it out and if you find the particular gene responsible for this or particular DNA responsible for this, you will be rewarded with Nobel Prize. That is why we are learning all these things. So this is the DNA which carries the heredity material from generation to generation. So whatever is there in a single cell, that is there within the entire body. Now, while some genes remains dormant, some genes remain active. So regulation of activities are there. Some regulatory mechanism are there. Thus we will learn in your NEET class that regulation of gene expression. What gene will express when? What gene will express when? That is the regulation of gene expression. We will learn. Now, for growth, DNA replication is required for growth, repair, and reproduction of an organism. This is the mission. Why? For the transmission of genetic information. It is required, DNA replication is required for the transmission of genetic information. For genetic information, transfer of genetic information, this DNA replication is required. So, whatever was there in a cell, that gene is perfect gene, peculiar DNA. That DNA has been conserved and that is being replicated in a very precise way, very serious precision. So, please give your time for 20 minutes to learn it. How it is done. Suppose DNA is a polymer of several nucleotides joined by sugar phosphate bond and other. We have learned it. Suppose if the sequence of a gene, DNA, let's say for example A, T, G, C, A, T, in this way the sequence are there. So these are the nitrogenous bases. Nitrogen is also an important item. These are the nitrogenous bases. They remain joined by hydrogen bond with thymine. These remain with adenine. This remain with 3 hydrogen bond. This is 3 hydrogen bond guanine. This is 2 hydrogen bond thymine. This is 2 hydrogen bond adenine. So we have seen that there are 4 types of nucleotides: A, T, G, C. Adenine, guanine, thymine, cytosine. These are the 4 nucleotides available. And this remains attached, they remains attached with the carbon 1 atom. This remains attached with the carbon 1 atom, carbon 1 position of sugar. This also remains joined with sugar. So I am writing sugar, sugar, sugar. Sugar. Then this sugar and remain joined with phosphate. Sugar remains with joined with phosphate. These also remain joined with phosphate. This is sugar phosphate backbone. This is the sugar phosphate backbone. We used to say this is the sugar phosphate backbone of a DNA. And the nitrogenous bases are attached with each other by hydrogen bond. This is phosphodiester bond. This is glycosidic bond, and this is hydrogen bond. This is the structure of DNA, and this is the structure of nucleotide. This is the structure of nucleotide. So there is some adenine, ESP. This is the structure of nucleotide. When 2 nucleotides remain joined, it becomes a polynucleotide chain or polynucleotide chain. DNA is the polymer of polynucleotide. And I have already given you information that there is 5' end and there is 3' end, which we have discussed earlier, need not to be go further. Now, this, once it breaks, once the hydrogen bond, say for example, this hydrogen bond is broken. So, this hydrogen is broken, this has gone to this side, this has gone to this side. This part is on that side, say this part is red, I will write it, I will give you red. This is sugar phosphate and T. This is the red one. Okay. And this one for differentiation, I am writing it in black. So this is this side and this is that side. This contains TACGTA, TACGTA, and this contains ATGCAT, ATGCAT. This is known as TACGTA. TACGTA. This has been separated, these two. So once it is separated, a young boy of class 2 or 3 or 4 or 5 can easily predict, we can easily predict that what the other, for replication, other he will come. band will be produced. So here you will see this red will be there and this black will be there. And this red will contains this T, A, C, G, A. T, A, C, G, A. And this will have A, T, G, C, A, T. A, T, G, C, A, T. A, T, G, C, A, T, G, C. A, T, G, C, G, C, A, T. So you can easily predict that whenever new cells, new DNA strand will be produced, that will contain the parent strand and the newly formed strand. The newly formed strand will have the same complementary basis. Suppose actually here what will be formed after sugar phosphate backbone. This one is red, I am writing is blue. So the sugar phosphate bond will have T will be having A. So actually this is being produced ATGCT, A, T, G, C, T will be produced. So it is the same identical copies. So DNA replication is the process by which the cell make two identical copies of its DNA. So these are the identical copies and these remain joined with this hydrogen bond. Again, this one will have, let me have some other color. This one will have A, T, A, G, C, G, A. Okay, with this sugar phosphate backbone. So easily we can predict that the new strand will be complementary to the parent strand. This is known as parent strand. This is parent strand. This is also parent strand. This is new strand. This is also new strand. So the new strand will have the complementary basis exact identical with that of the parent system, but this is new. Say for example, this is produced in 1965 and this is produced in 1966. So you can write that it is in 1966, it is in 1960 that it is produced. But both are same, both are identical. So DNA replication is the process by which the DNA of a cell, the cell replicates its DNA or made copies, identical copies of its DNA into two daughter DNA molecules. These two daughter molecules ultimately passes on to, in case of mitosis, it passes on here one, here one, here one, this one goes here, this one goes here. So it is replicate. And it is done so precisely, so minutely that there should not be any mistake. Suppose A mixes with T, here you write G. It can never be. Why? Anicoming. Actually, these two, that is guanine, cytosine, these two have definite length. These two are having a definite length, first of all, within the DNA. This is something 10.8 angstrom unit. 10.8 angstrom unit, the distance. So you cannot fit the other purine or pyrimidine bases other than this A and T. Thus, DNA molecule produces two DNA molecule. This is 11.8. Another one is 10.8. 11. This is 10.8, and another for GC the distance is 11.0. Somewhere it is written 11.1 angstrom unit. So distance is also precise. So you cannot fix other than purine and pyrimidine bases. Two purine bases you cannot fix. And even if from A it is a formula of ATGC, I told you on the other day. Remind it, ATGC is the formula. Having double bond, this is triple bond. You cannot mix A and C because one of these two are pyrimidine and another is pyrimidine. You cannot put another pyrimidine here. You cannot put another adenine here. This is the same precise engineering in the world. So many engineers has come. This is going on in my each and every millions and millions of cells in me. So we'll come just a little more 2 minutes. This is known as semi-conservative. It is conserving this, and this one is conserving the black one. So it is semi-conservative in nature. So DNA replication is semi-conservative in nature. Now, you have to prove, since you are science student, how will you prove? This what we are discussing, it is a discussion of history. It is a discussion of history only. No need of keeping in mind so many things. You should start. There was two scientists, one King, Watson and Crick, and King, two friends were there. In this way you can start. So Michelson and Stahl, what they did, two scientists, Michelson and Stahl is TAHL. Michelson and Stahl, these two scientists made a simple experiment. That simple experiment is nothing. These are the nitrogenous bases. This is nitrogenous bases. My name, bases. What they did it in some bacteria, the experiment. They put some nitrogen bases here, not bases, nitrogen salts. Nitrogen solution containing nitrogen. This contains 15N. It contains nitrogen 15N isotope. Simple, very simple, within 2 minutes I am completing. This contains 15N isotope. Usually the isotope of nitrogen is 14N. 14N is lighter, N, and this is heavier. So they have, what they did, they have cultured a bacteria, Escherichia coli, E. coli, some bacteria. So many bacteria they have taken, and they have incorporated. So it has got DNA replication. The DNA replication, where from these bases will come? It was the new bases. From the cell it has come. So these bacteria took the 15N nitrogen and produces nitrogenous bases. And after 2 or 3 or 4 generations, they have taken out this Escherichia coli, and this heavy DNA, heavy nitrogenous DNA, nitrogen 15N nitrogen contains DNA, can be differentiated by centrifugation. In centrifugal machine, you can centrifuge and take out this 15N DNA. So that 15N DNA bacteria, they are isolated. Okay. Thereafter, they did nothing. They put this bacteria in 14N. Nitrogen isotope 14N. It contains nitrogen solution, nitrogen salts of 14N. And they grew the bacteria here. They grew the bacteria here. One or two bacteria they have taken, isolated. Both of them have DNA of 15N. This is 15N, 15N. Because they have grown there in 2 or 3 successive generations, they have isolated those bacteria which have 215N DNA contents. So this containing 15N in their nitrogenous bases, in their DNA, in their polynucleotide chain. Now, after one generation, when the bacteria multiplicated into 2, so this bacteria multiplicated into 2, one is this, one is this, these bacteria, they have taken the DNA of this, taken the DNA of the and evaluates its density. Its density was evaluated. This can be done. So when they evaluated this, they have seen one strand contains, this one strand contains 15N and the other strand contains this 14N. The new strand has been synthesized, it contains 14N. Both are true. In case of here also, it is 15N and this is 14N. So it is a conclusive proof that the DNA replication is semi-conservative. This is the Nesselson and Steele experiment, and whatever I have said, that is the history part. So this may come, this is the simple question, it may come that explain the DNA, define DNA replication with the help of experiment. We will write up to these two. This will be the end of this episode. In the next episode, I will come into the DNA replication process, the actual process. It is a little bit difficult, not that much difficult that it is not a rocket science or something like that. We will be able to understand. And the next year we will do it. Till then, thank you very much. Stay healthy.